said, I'm a postdoctoral fellow at MGH, and I will say I have no disclosures, no financial disclosures. So today I'm going to be talking about targeting sleep oscillations to improve memory, and primarily looking at human subjects and human data, and really going over an intro of this relationship between sleep oscillations and memory, relating it to neurodegenerative diseases, but really going over data from the lab I'm in and that my PI, Dara Minowick, has done, looking at sleep oscillations in memory and schizophrenia. And hopefully throughout this, I'll be able to convince you all that there are a lot of similarities between cognitive and memory deficits in schizophrenia and neurodegenerative diseases, at least in relation to sleep oscillations and sleep-dependent memory consolidation. All right, so first off, I just want to go through this relationship between sleep and memory. And for most types of memory, we think consolidation as happening primarily during non-rapid eye movement sleep. So that's non-REM sleep, and specifically stages 2 and 3. So when you're looking at stage 2 and 3 of non-REM sleep, there are three kind of dominant oscillations. You have these cortical slow oscillations here that we've outlined in red. These are 0.5 to 2 hertz oscillations. They're generated in the cortex. And they have this characteristic shape where they have a down state, which is a hyperpolarized state. Cortical pyramidal neurons generally are quite silent during that period. And then they have this depolarized up state where you get a lot of firing in cortical pyramidal neurons. Next up, we have these thalamocortical spindles. So these are 12 to 16 hertz oscillations. They're generated by the thalamic reticular nucleus, which inhibits actually like primary thalamic nuclei, which actually project to the cortex. And so that's how these spindles, while they're generated in the thalamus, they are projected to the cortex. And the last oscillation here are hippocampal ripples. So these are generated in the hippocampus, deeper in the brain, and they're 70 to about 150 hertz in humans. And we know that these correlate with network patterns of activity and actual individual neuron firing of reactivation of experiences during wake. And this is rodent work done by Matt Wilson's lab at MIT and the Buzaki lab at NYU, among others. So one feature that I really want to highlight here, which might not be clear just from this cartoon, is that the spindles generally occur during the slow oscillation upstate, so this transition to the upstate, and generally peak during the peak of the upstate. And that hippocampal ripples actually occur at specific times of the thalamocortical spindles. So there's a coordination between these oscillations, and they all interact with each other. And we think what happens during these oscillations is that the memories are actually being consolidated from temporary storage in the hippocampus, where memories are stored during wake, to long-term storage in the cortex. And it's the coordination between these that allow this to happen. The other thing with this is that from EEG signals, so scalp EEG, non-invasive recordings, we can get these spindles and cortical slow oscillations. However, we can't get the hippocampal ripples with scalp EEG. So most of what I'll be talking about today is acquired through scalp EEG, and we kind of have to look at some of the rodent work as well as a little bit of the intracranial work in human subjects to try to look at the relationship between these, between all three of them. All right. So we also know that optogenetic and electrical stimulation studies in rodents and pharmacological and stimulation studies in humans have established sleep spindles, and to different extents, these three oscillations as a mechanism of memory consolidation and as a potential promising biomarker. And I think through this presentation, I'd like to show everybody that I think these sleep oscillations are a good biomarker, though there is, more work to be done with them. Okay. So sleep-dependent memory has been shown in humans across a wide variety of different types of memory. So for one example here, early work by Jessica Payne and our collaborator Bob Stickold found that kind of extracting the gist, or in this example, it's actually a false memory, improves with sleep. So you have a list of words, and people end up remembering a word that's not on this list that's associated with the words on the list. So window was never presented here in pink, but people say that they were shown window after they're shown things like door, glass, pane, shade, etc. And this improves with sleep. Other examples are emotional memory. Memory of emotional scenes increases with sleep. Maze tasks, so kind of a spatial memory, improves with sleep. Extracting insight, so extracting, say, a shortcut, not necessarily from a maze, but a shortcut, just like a stepwise shortcut, can be improved by sleep. And also language learning in children is facilitated by sleep. So sleep is really an active period for cognitive functioning and memory consolidation. And from these results in healthy subjects, the question then is, well, could abnormal sleep contribute to cognitive disorders? In our lab, specifically with schizophrenia, but also in neurodegenerative disorders, kind of disorders across the spectrum of various diseases. So what I wanted to start with was a little bit of an overview of the relationship in healthy aging and Alzheimer's disease, to give you a flavor for how this can be expanded across conditions, and then really dive into more work out of our lab. So there's a review by Mander in 2017 that was looking at just different sleep oscillations in healthy aging. And what they found in this review, what they went over was looking at young adults and older adults. That would be generally people in their 20s versus people in their 70s. They found there was a reduction in slow wave activity. So that's kind of band power in the slow wave band. And they also found a reduction in slow wave density. And density in this instance is talking about the number of slow waves per minute of sleep. So they found reduction in slow waves in aging compared to young adults. And they also find a reduction in spindles, specifically sleep spindle density in older adults compared to younger adults. So this has been known for probably 10 to 15 years. And more recently, work by Helfrich actually looked at the coordination of these two. So we have deficits in slow wave activity in spindles. How about their coordination? And the paper by Helfrich found that actually there's a difference in coordination between younger and older adults. So you have older adults on the left here, and the slow wave activity is in black there, and the spindle is in blue. And with the young adults on the right, you have the slow wave in black and the spindle in red. And what you see here is that in older adults, the spindles peak at an earlier phase. So they peak before the upstate of the slow oscillation, whereas in younger adults, they peak right around the upstate. And these are examples here. On the bottom, you have the distribution of those across the subjects, and you see very clearly that the older adults, the peak of the spindle is before the 0, which is the peak of the slow oscillation, whereas in younger adults, it is kind of, the mean is right at that peak. And what they found, maybe perhaps more importantly, in this relationship between memory and sleep oscillations, is that the phase of the coupling between the spindle and the slow oscillation actually correlates with the memory consolidation. So the closer to the peak the spindles are coupled, the better the memory retention. And this is true kind of differentially for both older adults and younger adults, where if you go off that peak, or if your spindles peak off of the slow oscillation peak, you actually have less memory retention. So that's with healthy aging, where there can still be some memory and cognitive deficits. When you look at individuals with Alzheimer's disease and mild cognitive impairment, you also see, compared to age-matched controls, that they actually have decreased spindle density as well. So decreased compared to younger adults, but also decreased to age-matched controls. And this group found that there's a correlation between the mini mental status exam, which is a measure of cognitive function, and spindle density. So the more cognitive dysfunction, the fewer spindles. And other groups have found this kind of throughout the age span, this relationship between kind of cognition and spindle density. Okay, so we have that relationship set up in at least one set of neurodegenerative diseases. So now I'm going to transition over to schizophrenia. And this might be a little less familiar to people in this group, so I want to go through this a bit more. So schizophrenia, generally the onset is in late adolescence or early adulthood. Drug development, so antipsychotics, which you generally think this is what is used for schizophrenia, is really focused on symptoms, and a lot of the time they're focused on these positive symptoms. So what you might stereotypically think of as something like auditory hallucinations. That's really what they focus on. That's something that is relatively easy to measure, and something that you know, has been a target of these antipsychotics for decades. But even after successful treatment of those symptoms, cognitive deficits still remain. And what this leads to is such things as individuals with schizophrenia, approximately 80% are unemployed. So, you know, that's quite problematic. And these cognitive deficits are the strongest predictors of functional outcomes. So being able to hold a job, being able to do activities of daily living, things like that. And with this, there are also, you know, staggering costs. So schizophrenia strikes when people are young, and there's money that goes into, of course, treating, but also, you know, a loss of jobs, you know, cost and toll on caretakers, all that. So yeah, there are staggering costs, and it's really an unmet need for the field, is really understanding how we can ameliorate these cognitive deficits. And we don't understand their neural basis. So hopefully, you know, looking at these sleep oscillations and understanding this relationship can help us meet this unmet need and develop effective treatments. All right. So with that, I want to dive into kind of the task, one of the tasks we primarily use for looking at memory consolidation in these subjects. This is the finger tapping motor sequence task. So it's a procedural memory task. We also use declarative memory tasks, but in this case, we'll be talking about procedural. And what we have participants do is type a sequence of numbers. Here it would be 4, 1, 3, 2, 4, as quickly and accurately as they can with their non-dominant hand. We have them do this 12 times for 30 seconds with a 30-second break between each block. And we have them do this before sleep, and then we have them sleep and do this task again. And we calculate the amount of improvement that occurs from the end of their training to the beginning of their testing. And this has been well documented in healthy controls that there is a jump in performance even without practice. And this jump depends on having a period of sleep. So if people are kept awake, they don't have this jump in performance. All right, so some early work in the lab looked at this just behaviorally. So what I'm showing here is the number of sequences per 30 seconds on the y-axis and the training trials on the x-axis. And in blue here is the healthy controls. So you see they have a nice learning curve, and they kind of plateau near kind of this 8 to 12 trials. But they have a pretty steep learning curve in these first couple of trials. This gray bar is a sleep period, overnight sleep period. And then we have the testing 12 blocks here. And you can see that there's this jump, and is 11% in the study from the last 3 training trials to the first 3 testing trials. Now I'm going to show the patients with schizophrenia. And here we have a different y-axis. So these cognitive deficits also are reflected in this procedural memory task. But what we see is that in the red triangles here, that these individuals with schizophrenia show a very similar learning curve. They don't reach the same plateau, but this trajectory is very similar between the healthy controls and the individuals with schizophrenia. But then when you look at their performance in the testing period after sleep, you see that they don't show this overnight improvement. They actually show a slight decrement here. Yeah, so there is this large difference in this, specifically in this sleep-dependent aspect of this. So this is kind of the basis for the work we're doing now, and the basis of the lab for the past 15 years. So then the question is, we have a behavioral change. So what in sleep correlates with this? So in a separate independent sample of patients with schizophrenia and healthy controls, postdoc in the lab and a collaborator's lab actually looked at this in patients with schizophrenia compared to the healthy controls. And looking at electrode CZ, so that's kind of in the middle of the head, they find that there was a decrease in spindle density, overnight spindle density in patients with schizophrenia compared to the controls. So this is very similar, again, to what you're seeing in healthy aging, but also in AD versus healthy older adults. What they found is that, again, in these patients with schizophrenia, the spindle density correlated with the oversleep memory improvement. So the greater the spindle density overnight, the greater the memory improvement. All right, so let's see. Okay, yes, moving on. So there is a spindle density aspect, but of course, I started this by talking about the coordination between oscillations. So moving on from solely spindles, and this is kind of where the field is going, rather than looking at specific one single oscillation, looking at this coordination. In another set of subjects, Charmaine de Manuel, who was a postdoc in the lab, again, was looking at the relationship between these oscillations and memory improvement in schizophrenia. And she found that spindle density, as they found with the previous group, correlated with memory improvement, but also that the slow oscillation spindle phase consistency in patients with schizophrenia correlated with this memory improvement. So that coordination aspect and combining them together was an even better predictor of memory improvement. So I'll leave it at that in terms of this relationship with coordination, but it's something to think about that I'll go to after this. So the question then is, can we intervene on this? Other things, are there different interventions that might be able to increase spindles, increase slow oscillation, improve sleep, and hopefully improve memory? And not surprisingly, perhaps, the answer is yes, there are ways to do this. So I'm going to outline three here. So one would be a pharmacological approach. So it's been long known that, for example, benzodiazepines or just GABA receptor agonists can increase spindles. So that could be one approach you could take. Another one could be electrical magnetic stimulation. So this is slightly more recent than the pharma approach, but using transcranial current stimulation or transcranial magnetic stimulation, we might be able to target some of these oscillations. And different groups have looked at different aspects of that. And I can talk more about that if people are interested. And the other approach, which is one of the ones that we decided to do and are currently working on, is doing sensory stimulation. So if you play stimuli during sleep, you can actually evoke some of these oscillations. And I'll go through that a bit later. So the early work in the lab, looking at this spindle relationship between spindles and memory, and then coordination and memory in patients with schizophrenia. My PI and other members of the lab, showing two of them here, decided to do a clinical trial using isopuclone, which is a GABA A receptor agonist. So it's a sedative hypnotic agent. And using isopuclone, which is known to increase spindles, we found that here we have healthy controls on the left, schizophrenia patients on the right, placebo and isopuclone. Find that isopuclone increases spindle density. So you can increase spindles with isopuclone, which, as part of the story is, I think, quite important. And what they found also is that, again, the spindle density with both placebo and isopuclone correlate with MST improvement. Again, kind of reiterating earlier results. But they found no improvement in the motor sequence task in either the controls or the patients with schizophrenia. So then the question is why might that be? And what Dimitris found was that isopuclone also affects slow oscillation. So, one of these things I think with pharmacological agents is that, they can have system-wide effects. Though, of course, perhaps they can be targeted as well in a very specific way. What they found with isopuclone was that isopuclone actually decreases the slow oscillation amplitude compared to a placebo. It also decreases the consistency of coupling between the spindles and the slow oscillations. So, as Helfrich found in the healthy aging, that consistency is important with memory consolidation, as we found in schizophrenia. So isoprolone decreases both of these. However, we also found that the coupling density correlated with MST improvement. So, you know, a positive kind of marker that really maybe this coupling is a better predictor than spindles alone. And a last part of this is that the amplitude of the slow oscillation actually correlates with the consistency. So really kind of reiterating that if you're screwing up the slow oscillations, you can screw up this coupling and in turn, you know, not improve memory. So we have to keep in mind that the coupling, the timing of the coupling really is important, and we need to maintain that. All right. And the last part of this is, of course, the hippocampal ripples, which we didn't get to in humans with this, but luckily we have collaborators. And here we're showing Carmen Varela, who's at MIT, Matt Wilson, who luckily have rodent models for this. So what they can do is record from the prefrontal cortex, the thalamus, and CA1 or other areas of the hippocampus at the same time, and look at how these drugs actually influence, you know, the spindles or the ripples here in the hippocampus, and use that as a, you know, this animal model of what might be going on in the humans who are taking these drugs. So they did these studies with isoprolone as well as zolpidem. So zolpidem is another GABA A receptor agonist that has different binding properties. And what they found in the rodents, and here we're just looking at the hippocampal recording and looking at a vehicle and isoprolone, and this is looking in the ripple band. And what you have on the bottom is x-axis is time in minutes. So you're giving the vehicle or isoprolone and you're looking at, you know, the ripple power. And what you find is that isoprolone actually also decreases the ripple band power, so probably inhibiting ripples. However, if you look at zolpidem, you know, another agent, you can actually see that it increases the ripple band power. And when you look, you know, averaging over all this, you find, you know, with just the vehicle, you know, normalized to 1. And then with isoprolone, you get a decrease, and zolpidem, you get an increase. So, you know, we initially were using isoprolone. Now, based on these studies, we're doing a clinical trial using zolpidem, which hopefully will increase the coupling of these oscillations. And from this study, you know, also increase those ripples to improve memory. So that's an ongoing study that we're recruiting and running right now. All right. So switching over to another potential intervention is this closed loop auditory stimulation. So what this consists of is recording EEG from a sleeping participant. We filter the signal as slow oscillation band, this 0.5 to 4 hertz, and we have an algorithm that detects when the downstate of a slow oscillation occurs. And then we play a quiet sound, not to wake the subject up, but to actually evoke this brain response. And what we see is that we can evoke another slow oscillation when we play this quiet sound during the detected slow oscillation upstate. And in this example here, we also evoke a spindle. So this would be a coordinated slow oscillation spindle. So why, you know, this work, I would say, has been known for a while, that you can play or you can deliver stimuli during sleep and evoke these. Why do we think they can improve memory, besides this kind of theoretical model of memory consolidation? Well, a group led by Ngo et al. In 2013 published a study where they were doing this closed loop auditory stimulation. And for their specific setup, they were doing 2 stimuli at subsequent slow oscillation upstates. What you see here in B is the slow oscillation amplitude on the y-axis, time on the x-axis, and these dotted lines are when they delivered the sound, or in the sham, when they detected the slow oscillation and would have delivered the sound. And you see that with stimulation, you do evoke additional slow oscillations. What they also found was an increase with stimulation, an increase in this fast spindle power, this fast sigma band. They didn't detect these oscillations discretely, but they did see an increase in these frequency bands of interest. And they also found an increase in retention with stimulation. So that's very promising. Other groups have replicated these results. So we decided, we're looking at patients with schizophrenia. We wanted to get this set up in our hands and see what happens starting with healthy controls. So this is some more recent data that we've collected. We were doing the stimulation. This is set up, very similarly to the previous graph, where we have the amplitude on the y-axis, time on the x, and stimulation versus detection in red versus black. And we decided to do one stimulus at a time so that we can really understand more what's going on after the stimulation. And we find with stimulation, this is with 20 healthy controls, we can evoke slow oscillations. When we look in the sigma band, we found that we also increase the sigma activity, and the peak is near the peak of the slow oscillation. So based on this, based on the power in these bands, we're increasing, the target of what we want to increase. But we actually wanted to detect these oscillations because we think this, and we found this, these discrete oscillations actually correlate with memory. So here I'm showing the probability of evoking these oscillations. So starting with the slow oscillations with no stimulation versus upstate stimulation. And what we find is, kind of as you could see from this graph, we definitely increase the probability of evoking oscillations compared to no stimulation with these auditory stimuli. And that's basically across the entire brain. If you look at spindles, we do increase the probability of evoking spindles primarily frontally here with the stimulation. And if you look at these slow oscillation coupled spindles, you see that we also increase the probability of evoking these axes or the intensities are different probabilities. So we have the greatest probability of evoking slow oscillations, and it kind of decreases with this, but we still have a greater probability of evoking the slow oscillation coupled spindles frontally, and also medially going all the way back to the parietal electrodes. So we don't see a drastic effect on memory in these healthy controls. We do see kind of a trend towards improved memory with stimulation. But we also wanted to look at the correlation between these oscillations and the density of these oscillations over the entire nap. So not just stimulated ones, but over the entire nap, and MST improvement, because we wanted to replicate these results we were finding in our previous studies and that other groups have looked at. And what we found in our healthy controls is that the slow oscillation density and spindle density don't correlate with this memory improvement, but actually coupling density does. And we had the subjects here, I'm showing the 3 sessions. We had another session of stimulation where we stimulated at a random time to emphasize that timing is important. Not going to get into that now, but I'd be happy to talk with people more about that. But we found across these 3 sessions, there's this really quite strong relationship between coupling density and MST improvement. So that's, I think, very promising. We are currently doing this stimulation in patients with schizophrenia, having them come in, do the task, take a nap in the lab, and then they're tested on the task. And I'd say preliminarily, at least, we are able to evoke these oscillations in patients with schizophrenia. I have not looked at the memory results yet, and that's kind of an ongoing project we're doing. So bringing it back to kind of the neurodegenerative disorder side of things, there's a group in Northwestern that was also looking at this in healthy aging and patients with mild cognitive impairment. And what they found, just in brief to bring it back to that, is that with their stimulation, they increase the slow wave activity. So they're increasing slow waves in healthy older adults and also older adults with mild cognitive impairment. And they were kind of trending towards an increase in this fast spindle band or the sigma activity over here. So, even in older adults, you're able to engage this target, maybe slightly differently than in younger adults. And kind of that speaks to an optimization problem with this and trying to figure out really how the networks are responding in different people and in different populations, which is, I think, the next big task that we have for this. And they also found that the changes in slow wave activity correlated with their overnight changes in memory improvement in both healthy adults and those with mild cognitive impairment. So again, really hitting on this relationship between the oscillations and memory. And then kind of the last part of this stimulation story is that we're really, I think, excited about it because it can also be done in the home. So, which of course, pharmacology, people can take drugs in their home as well. But this isn't something that needs to be held in the lab and can only be done in the lab. There are devices that have been developed to do this closed loop stimulation, recording from EEG, having an algorithm to detect when the slow oscillations occur and actually triggering the quiet sounds during sleep for home use. And so that's I think, very promising and really opens an avenue for doing larger and potentially longer term studies, because sleep studies are, quite resource intensive, and being able to, send somebody, say, home with one of these and they wear it for a week or, a month or whatever it might be, to really collect that data and understand how the stimulation is affecting things like sleep quality, outside of these oscillations, I think is very promising way forward with this. So the last part, and this is kind of my last data slide, is, in all these studies, except for the rodent ones, we're really missing the ripple component, that third oscillation that we think is really coordinating this memory consolidation. And so this is a project that we've started working on with our collaborator, Catherine Chu, who is an epileptologist and neurologist at MGH. And what we're doing is actually looking at these oscillations and the stimulation in patients with epilepsy who are coming in to the epilepsy monitoring unit because they have treatment resistant or treatment refractory epilepsy. And so they're coming in pre-surgically to get the seizure foci localized. So the neurologist and the neurosurgeon plan where they're going to put electrodes, intracranial electrodes, into the brain of these patients to try to figure out where their seizures are coming from for a potential neural stimulation device or potential resection. And we're kind of piggybacking, collaborating with them to run these studies in these patients to get this ripple activity that we think is so important. So here I'm just showing an example of an MRI with a CT image of the electrode superimposed on it. So you see that there's different leads here across the brain that are, again, all clinically targeted. And I have one contact here that's in the left hippocampus, contact 2 that I just want to show we are getting, hippocampal coverage and hippocampal recordings. And what we can see, this is data from one subject, and it's time locked to detected ripples, is we get a very clear island of ripple activity around these detected ripples. So we can successfully detect them, and they're in this 70 to about 120 hertz band that is the range of human ripples. And here, kind of the last part of it is, we have 21 electrode scalp EEG coverage on these subjects, as well as the hippocampal EEG. In black here, I have the slow oscillation. In red here, we have a spindle. So this is a coupled slow oscillation spindle event. And what we see in the hippocampal EEG, in dark blue is a wideband filtered signal, and in light blue is the power in just the ripple band. And what I'm showing here is actually a triple coupled event. So we can get that in these subjects. In a spindle with these, you see these different cycles of a detected ripple here, and you see this increase in the ripple power. So I think it's very promising doing these simultaneous kind of non-invasive studies with EEG, memory consolidation, and then using these studies in intracranial subjects to really tease apart how interventions could coordinate these oscillations and how that can hopefully improve memory. All right, and with that, I'm open to any questions. Of course, I want to thank everybody in the lab that's collected this data, been part of the discussion, collaborated with us to do all these analyses, and of course funding sources, because otherwise we couldn't do any of this science.